So main functions of blood, we can talk about blood as a whole, all the different things, right? So blood as a whole will transport things, right? In terms of protection, it's fight against foreign organisms. Which part of the blood does that? White blood cells, right? Clotting,
Platelets.
that's the platelets, and we'll talk about other factors in our blood plasma called clotting factors. Acid-base balance, we have a buffer in our blood. Who recalls what's the name of the buffer in blood? The bicarbonate buffer. Fluid electrolyte balance, it balances fluid in the in our cells, within our cells. It takes it out and puts it in, depending on osmosis, right? And electricity. Regulation of temperature, you can talk about how blood will carry heat. So if we close off blood supply, we're going to carry less, hence lose less heat, right? During cold, you'll see the blood supply to especially the skin, you can notice as heat decreased, where if you're feeling warm, hot, it'll actually increase, trying to get rid of temperature or heat in our body. So let's talk about the blood composition. If we leave it or we spin it, it's going to separate out. Okay, it's a suspension. So at the bottom is going to be the cells. Bulk of that cells, and we'll talk about the numbers in a little bit, is red blood cells. So 45% by volume of blood is occupied by red blood cells. And that has a name, that's called hematocrit. Okay, hematocrit is basically how much of the volume of the blood is occupied by the red blood cells. Okay, so it's like 45%. Okay, above that we have what's called the buffy coat. That's going to be the white blood cells and platelets. And then blood plasma, I know it doesn't add up exactly to 100%, it's rough estimates, right? So this is less than 1%, but 55% of the blood plasma, that's extracellular fluid, right? This is not inside cells. Really, these three things, we don't really like using the word cells. One is not a cell at all. Okay? The other one cannot replicate, it does not have nucleus. So the better term is form elements. Okay, that's going to be our erythrocytes, that's our red blood cells. We'll talk about serum in a second. And then leukocytes and thrombocytes. Thrombocytes are not cells at all. They're pinched off pieces of cells. You may have heard of serum. It's similar to plasma but different. How is it different? So if you remove the clotting factors from blood plasma, that's going to be serum. Who may receive plasma transfusion? What's contained in blood plasma? Besides water? Clotting factors. So if someone who has a bleeding disorder, they're bleeding, they're not able to clot, we may give them fresh frozen plasma to replace those clotting. We also give them to liver patients. Why? Plasma, fresh frozen plasma. Yes, it could be for the clotting factors if they have decreased clotting factors. Where are clotting factors made? It's the liver that makes them. But it's another thing that the liver makes. That's found in the blood plasma. It's albumin. Right? It's a protein, so keep it that too. Well, where do we give serum? We did that at the start of COVID infection. That was our only treatment initially. What was it that serum? Antibodies, right? So somebody who had COVID, recovered, we took their blood, we took out the antibodies, we transfused. Right? We've heard of antivenom. That's antibodies towards the snake. Right? So those are serum. There are others too we use. So serum is basically plasma minus the clotting. Okay? But we generally take them or use them for antibodies. So let's talk a little bit about them. Red blood cell is the most numerous, right? They're in the millions, God bless you, per microliter. They're about 4.5 to 5, or really 3.5 to 5 million cells per microliter, right? Next is the platelets. It's in the hundreds, okay? Hundreds of thousands, rather, should I say. It's about 150 to 500,000 cells or platelets per microliter. Right? While white blood cells are the least numerous, they are in the tens of thousands, really. They're between 4 to 10,000 per microliter. While in the blood plasma, we have many things. I mentioned about water. We do have electrolytes dissolved there. We have nutrients, glucose, amino acids, right? We have some waste products like urea. Okay, what is the urea from? Got rid of it. It's from breakdown of proteins. It's the ammonia part. Because of the toxins for us. That's carried, again, it can be carried it's less toxic than ammonia itself. Okay, so it's carried, but again, if we have too much urea, uremia, that will cause bad, will cause mental status changes and others. We also have plasma proteins. The most common, or 60% of the plasma proteins, is albumin. Okay? That's the most in of plasma proteins. While these globulins, these are a group within which immunoglobulins or antibodies are also found. That's what we will take out. Okay? Then what is fibrinogen? Yeah, it's the last stage in clotting. We'll talk about how fibrinogen becomes fibrin. Okay, that's coagulation. It's going to form this net, and everything, including platelets, but also white blood cells, red blood cells will get stuck. That will form nice stuff in the and then we have other proteins there too, like enzymes, hormones, right, found in blood plasma, the liquid portion, the extracellular matrix of blood. And we'll talk about the different types of white blood cells. We have 5 different white blood cells in lab. We'll should be able to identify them, and then importantly also know their functions, which we'll talk about today. So let's talk about some terms in terms of blood collection and analysis. The one that is regularly done is venipuncture, right? So we're taking blood from the veins. We can run a number of different tests to check how much of the white blood cells, red blood cells, platelets they have. We can check the electrolytes, we can check clotting factors, how quickly they clot, and so on, right? What's our favorite blood vessel that we like to take it from? Please don't say it, right? Well, say what you want, and then we might have it corrected if you don't know the exact one, but usually people say where it is, but not where it is. What's that one? AC vein, yeah. That's AC is the region. Antecubital region, right? I mentioned that the vein cell will learn it later in our blood vessel labs, median cubital. Okay? But it's in that area, hence, yes, if you know it, a cannula was put in the AC. Really, AC region is not the AC vein. Okay, that's the usual spot. Yes, we can take it from other places. We could take from jugular, we could take from femoral vein. Usually goes to the artery, so be careful right next to it. Jugular, we only use that for central line, for example. It's something that we use in ultrasound. It's not usually done in the field generally. Capillary blood, we take it from the periphery. It was done as a tool, not necessarily, or very commonly for glucose levels, right? We can also test it if you go to donate blood. Where will they take to check your hemoglobin levels?
Your finger.
It's the capillaries again, it's the fingertips. Okay? We also can check INR in the fingertip too. What's that done for? A lot of things to do with, especially if they're on anticoagulants. What are some anticoagulants? Coumadin, warfarin, we need to check INR, right? We can do that. And generally from the fingertip, but there is other places also possible. Infants, we don't do the fingertip, we actually do the heels. But again, it's capillary, take a little bit, it still can give us information, right? Then the other one is arterial puncture. Again, not usually done in the field, but in hospital setting, we take blood from an artery, right? Is this painful? What do you think?
Yes.
Yes, it is. Okay, because artery is carrying blood under high pressure. We thought veins when you donate blood, they can be painful because they do put a large blue cannula in there. But this is even more painful because by adaptation, our body is trying to protect it, right? Because what is pain again? It's a warning about
Cell damage.
damage or imminent damage, right? Hence, we have a lot of pain receptors around it. So we can, usually we go here, we can, not very often there. Again, that could be done in ICU, groin, we do take. This is usually for blood gases, that's usually what it's done. And then we'll talk about evaluating the amounts of the pH, the bicarbonates, and called partial pressure of CO2. You can also check the partial pressure of oxygen too. That's usually checked. Radial artery is the most common. Where's the radial artery? Yeah, if you feel the pulse, that's what we usually use. And that's the most common one we use. It's on the radial side, thumb side. Right, that's what we will use as well for blood gases. And we've mentioned arterial pH of blood is 7.35 to 7.45. Okay? That's the range within which blood pH to be looked. So this is very small. This kind of goes through what we talked about in terms of proteins. Here it elaborates. We'll talk a little bit more, not a lot more, about these globulins. We will talk about fibrin and other clotting factors here. Just mention these ones, right? We talked about how water is the bulk of the plasma, right? But with other things dissolve, electrolytes, hormones, nitrogenous substances which are not protein-based necessarily, organic nutrients, and then the respiratory gases. What are respiratory gases? Oxygen and CO2. So we'll talk about them in the respiratory chapter. Yes, some is going to be attached to hemoglobin, but there's some in our blood plasma too, at different levels for the two different molecules we'll talk about. By the way, IF is interstitial fluid. That's also extracellular fluid. Do you recall where interstitial fluid is? It's between cells, right? The areola connective tissue has a lot of interstitial fluid. Okay? It's different from blood plasma and proteins. Interstitial fluid has virtually no proteins, otherwise it's very similar. Okay? Again, it's extracellular fluid, similar to our blood plasma. In terms of the proteins, most of the proteins are synthesized by our liver. Hormones can be synthesized by the different organs, right? Albumin, fibrinogen, globulins, it does some from here, some from our immune-produced immune system. It's our activated B cells which will produce the immunoglobulins antibodies as well. So in liver disorders, we can have all side effects because of the lack of uncontrolled bleeding, because the clotting factors that I mentioned there. They could have, we'll talk about this, albumin is like a attraction for water. So water stays in this compound, albumin is present. So when like a liver patient, they have less albumin, the water will not stay as much in the blood vessel that leaks out. So you can have lots of ascites, for example, like interstitial fluid collections, right? And legs are going to swell up because of the fluid leaking out as well. Right? So let's talk about hematopoiesis, different from hemopoiesis or homeostasis. But here it's the production of blood cells, hematopoiesis. You may or may not know the stem cells give rise to all types of blood cells. Okay, the one that has been least differentiated is this hemocytoblast. They give rise to all the white blood cells, the red blood cells, and the platelets. Okay? Then they separate out from this, will go into what's called lymphoid stem cells, and then the myeloid stem cells. Lymphoid basically just produces one type of white blood cells, the lymphocytes. Everything else is produced by the myeloid. You may have heard of myeloid in cancer conversation. What is that? What kind of cancer? Leukemia, myeloid leukemia. That tells you which line the cells are coming from. Lymphoid leukemia coming from the lymphocyte, lymphocytic stem cell line. Okay. Acute, chronic is how mature. Acute means they're very immature. None of them are functioning, right? Leukemia, they are producing these white blood cells. I put them in inverted commas, are not really proper white blood cells, they're non-functional. They're churning around a lot. Right? So we can classify by which line they came from, type of leukemia, cancer of the blood. So in terms of hematopoiesis, while we were still very small, the first 8 weeks of life was a yolk sac that produced it. Then the liver and spleen. Well, once we are born, it is the red bone marrow. In adults, where can we guarantee to find red bone marrow?
Flat or irregular bones.
Flat or irregular shaped bone within the spongy bone of those, right? Sternum, skull, vertebrae, and then the pelvis, which is what we donate from. So here's a picture trying to show that. That's the hematocytoblast that will give rise to myeloid stem cell and the lymphoid stem cell. The lymphoid stem cell basically gives rise to lymphocyte, while the myeloid gives rise to the others, including red blood cell, platelets, and the other 4 white blood cells. Okay. So let's talk about erythrocytes. What are erythrocytes?
Red blood cells.
Red blood cells. They're the most abundant, right? They're between 3 to 5 million cells per microliter. Okay? They are the bulk of the form. So in terms of hematocrit, this is the percentage of blood volume occupied by blood. As an average, we say 4 to 5 is considered the normal, right, as a benchmark. It can vary, right, in menstruating individuals. That can be lower, and that can be normal for them. It's fine, that is more than fine. While others can be high as 54, 55. Okay. Lifespan of a red blood cell is just about 4 months. Okay. Previously, we would donate and we'd wait 4 months for it. Okay. Now, how often can we donate full blood? Whole blood. We know.
Every other month.
Every other month, 8 weeks, right? It's about half that, because not all the cells are at that same stage. Some of them are almost at the end of their life, right? It's a variant, so hence it was found that was fine. And how much of blood do we donate?
1 to 2 pints.
Generally 1 pint, right? Because it's again, roughly how many pints of blood do we have flowing through our blood vessels?
6.
That's about, it's a little bit higher, it's about 8 pints, it's about 4 liters of blood. Okay? So we're basically giving up, if we give 1 pint, almost 15%. So we give too much, that you don't want to, it drops it. When you're giving double blood or double red cell, they actually take only the red cells, they give you the rest. They just don't take up 2 pints per se. That was still take less than that. Actually, they take less than a pint there when they take double red cells. But we can donate components to them. Usually, the mobile ones only can do whole blood or packed reds or double red cell. When we say we received or transfused blood, what do they really transfuse? Is the whole blood? Anybody know?
Usually the packed blood.
Yeah, it's the red blood cell. They say it's packed red blood cell. It's just a red blood cell that take the platelets out, white blood cells, as well as blood plasma. Not all of it, most of it out. A little bit of plasma has to be there for the cells to be present, but most of the plasma is removed and then we transfuse it for in the red blood. When the red blood cell comes to low, right? When it's bleeding from any reason or anemia or other causes. And we can give platelets only, and give plasma only as well. We have to go to center, either Kingston or the one in Fishkill. So they are biconcave shaped. Do they have a nucleus? Did we see them on the first day? We did see them in the histology lab, right? They do not have a nucleus. They've lost their nucleus. What do they contain? Hemoglobin. They actually have 250 million with a B molecules of hemoglobin. There's no nucleus, so they cannot replicate. So mature red blood cells lose most of their organelles. Nucleus is gone, even mitochondria is gone. Why might that be an advantage?
They have more space to carry more like oxygen, waste, and other nutrients.
Yep, so them not having other organelles is useful, but why not keep them in?
You don't want to use the oxygen that is carried.
Yeah, it's like ordering a pizza and the pizza delivery person eats some of the slices. Right? That's not going to be useful. Right? But can we still produce ATP without oxygen? What's that called? Anaerobic respiration. It's not a lot from each glucose. Does anyone know how many we produce? Just 2 ATPs from each glucose. A little bit longer. Okay. So it's by anaerobic because the main function of our erythrocytes is to carry oxygen. Okay, so hemoglobin is what the oxygen binds to, part of the hemoglobin. Okay, so 250 million molecules. Each hemoglobin molecule can bind 4 oxygen. So basically, one erythrocyte can carry 1 billion molecules of oxygen. Okay, they can carry some carbon dioxide. We'll talk about them. Most of the carbon dioxide is carried differently to here. It's only about 20% of the carbon dioxide is carried in the hemoglobin. 98% of oxygen is carried bound. Okay. So each one, each heme group has these 4 components, 2 alpha, 2 beta components. Each in the center, we have the heme group, right? Globulin, hemoglobulin, that has to do with proteins, right? So protein, 4 protein molecules with a hemoglobin each. So here is trying to show that 2 different kinds. That's what I'm showing. And then this disc would be the heme group. At the middle of the disc is, to which oxygen binds, is iron. Iron. Okay, that's where it binds to here. That's why iron is a raw material for building hemoglobin and red blood cells. It's basically 4 ions placed in each hemoglobin. So let's talk about the production of red blood cells. We have a hormone that will stimulate that. That hormone is erythropoietin. That's released by our kidneys when they sense that there is less oxygen in blood. Okay. They don't know if we are anemic or not. They have no sense of the number of red blood cells. They just know it by how much oxygen is being carried. Generally, it reflects it, but not necessarily, right? What's the percentage of oxygen in the air we breathe here? 21%, right? What's the Okay, I'll wait. What about up on a mountain? Right, we normally say that's different up there. What's different about it? Is the
Dependent.
percentage of oxygen different? It's the same, it's 21%, right? But the content is less, it's less molecules there. So we talk about pressure, higher here, pressure is how many molecules are hitting it. So we have a higher pressure with more molecules down here compared with higher, right? Still, percentage is still the same, 21%, but we have total less oxygen, okay? That can stimulate erythropoietin release. Because there's less oxygen in the air, how much oxygen is being carried by the blood?
Less.
It's going to decrease, right? So when it gets to kidneys are going to release erythropoietin. And what does erythropoietin do? It's going to stimulate our bone marrow to produce red blood cells. Okay? It does take some time, okay? Which is why if you're going to hike up Mount Everest without the mask, you have to go and stay on base camp for minimum 2 weeks, ideally 4 weeks, for your body to get adapted, and then because otherwise you will not have this. This increases the oxygen carrying capacity, basically, which has been used in sports to kind of cheat. Okay, blood doping is a little different. Does anyone know what blood doping is?
When you give yourself more red blood cells,
Yes, but where does that red blood cell come from?
what do like the transfusions?
Yeah, it's actually their own blood. So they take some blood out, they store it, what's happening to that person's body. It's going to stimulate red blood cell production, they're going to come back to normal, they put this other blood that they store, they generally store their own blood so they don't have problems of any transfusion defect of blood doping, their own blood, they transfuse it back. Now they've got extra red blood cells, extra oxygen carrying capacity, right? Some people cheat by giving themselves erythropoietin too. They run them in shots. What kind of patients receive erythropoietin? Maybe cancer, but it's gonna be, because kidneys are the one that produces it, they will not produce. It's usually given in erythropoietin kidney patients because they're not producing. Okay, so they can use artificial, which is called EPO for short, erythropoietin, which can increase. But when it increases, what part of the blood content is increasing? Red blood cells, so the hematocrit count goes up, right? Will that make the blood thicker or less thick? Thicker, so they are at a higher risk for developing blood clots because it's too packed. Okay? And it can be a dehydrated, that would augment it, so that can be very dangerous. So on the kind of the other scale is anemia. Please, if I ask you what is anemia, please don't say iron deficiency. That's only a type of anemia. Anemia is basically a low erythrocyte count. Okay? Low erythrocyte count. So reduced erythrocytes means reduced hemoglobin, which means reduced oxygen carrying capacity. Okay? They will experience weakness, fatigue, exertional dyspnea, so even slow movement will make them tired because they just don't have enough oxygen being carried in their body. Dyspnea means. So let's talk about some of them. I've listed 6, I believe, if I'm not mistaken. So if I ask, what are some types of anemia? Please list them this way, don't stop at iron deficiency. Okay, so these are some of them. Thanks. I'll explain them, what they mean. So if you ask for explanation, you also know that. Hemorrhagic. Hemorrhagia is to bleed, blood loss. So here it's something that's causing blood loss, whether it's a cut, whether it's menstruation, whether it's an ulcer, right? Something's causing blood loss. Okay, and the red blood cell count decreases, it's anemia. This is hemorrhagic anemia. What is lysis? We talked about hemolysis somewhere else. Lysis or lysis?
It's when the cell bursts.
When the cell bursts, right? Good. So hemolytic anemia, the next one here, the red blood cells have been broken down for a reason. right? For example, sickle cell disease could result in anemia because these cells are going to be, some of them will be just broken down. If we transfuse incorrectly, our body will break down those cells as well as our own red blood cells too. Okay? Transfusion issues can cause it. Some infections can trigger it as well. Thalassemia is a type again of red blood cell disorder. Sickle cell. What's the sickle cell disease? Yeah, they're more crescent shaped. Sickle was a instrument that farmers used for harvesting. It was the shape of a crescent. With a metal, a sharp. That's where it came from. How does it come to be? What's the reason for sickle cell? Question or answer?
Answer.
Sure.
It was a failed adaptation for malaria, so people in the Middle East and African areas developed these cells to...
Yes and no, I'll come back to that in 1 second. Yes, it's more common in those areas. We'll talk about that in 1 second. But what's different about this compared to a normal red blood cell? Shape. Why? It has to do with one amino acid in the protein sequence of the human. So that's all. It was one sequence. And normally we get a difference in amino acid. There's a mutation in the DNA. Okay? Mutations are chance mutations. Whereas what you talk about is talking about how evolution made it so that in areas that were malaria bound, there was a higher rate of sickle cell, right? Because malaria is it's plasmodium, it's a microorganism that lives in red blood cells. It cannot live in sickle cells, right? So, does anyone know how malaria spread?
Mosquitoes.
Mosquito bites, right? They transfer from an infected blood to another person. Okay, so these will destroy red blood cells, the malaria does. But sickle cell, individuals with sickle cell, either trait or disease, we'll talk about that in a second, they were more immune to that, right? So an area where there was more malaria, they survived, while people that were normal blood, they did not. So when the next generation, again, do we individually evolve? As individuals, we do not. It's the population that evolves, right? It's a change in the genetics makeup of it. So who survived had sickle cell disease, they reproduced, next generation and so on, multiple generations later, we found there was a lot of individuals in that area who didn't have this trait or disease. Trait basically means that one gene for it. Disease is when they have both genes, you actually see the proper disease of the cell. It's a trait, you can lead to it. They have sometimes they produce those because depending on which chromosome we trigger for the production. So they may have some cells which are sickle cell. Anyway, that's hemolytic, so those will be broken down. Okay, that can lead to low blood cells. Next one's aplastic. Here it's in the process of producing this product. Okay, so it's usually a bone marrow issue, either destruction of bone marrow, intentional or otherwise, or a suppression. For example, if I was to receive a liver transplant, what would I be put on? Yeah, anti-rejection medications. What are they?
Immunosuppressants.
Immunosuppressants. When they suppress our immune system, we're talking about the bone marrow. Okay? One of the side effects is that I may develop anemia, so I'm not able to produce because my bone marrow is not being stimulated. Okay? That could cause, that's one of the drugs that can cause it. Radiation, for example, for leukemia, treatment is chemotherapy and radiation, where you are basically wiping out their bone marrow, which is the one that's producing the leukemia, right? Side effects, yes, we've gotten rid of the cancer, but we've also gotten rid of their ability to produce the blood cells, right? It could be a side effect of other radiations. Next one is something called pernicious anemia. Here, I'm going to track back to produce red blood cell, we need B12. Okay, B12 is absorbed in our small intestines. Even if we consume B12, if there's another molecule not produced, B12 is not absorbed. That other molecule is called intrinsic factor. Does anyone know where intrinsic factor is produced? It's not in the small intestines. It's actually in the stomach. So somebody who has gastric bypass surgery or has removal of part of their stomach could suffer with pernicious anemia because they may not be producing sufficient intrinsic factor. Okay? So the problem is not, even if they consume B12 enough and more, they still will not absorb. Okay, because the treatment is placing it into the blood directly, either into the blood vessels or through iron injections. So pills usually are not good. And then we also have iron deficiency. So here we have one of the important raw materials for developing hemoglobin or making hemoglobin is missing. Right? Poor absorption or not enough in the blood. Another one is the renal anemia. This one I put it because we talked about erythropoietin, right? So in kidney, end-stage kidney patients, they do not produce enough erythropoietin, hence do not stimulate their red bone marrow to produce red blood cells. Right? So for them, we'll just give them injections, usually a monthly injection of erythropoietin to stimulate their red bone marrow to produce to produce the erythrocytes. Questions? While polycythemia is the opposite, when we have more red blood cells. Okay? That term, yes, it may translate to more cells, but it's really more red blood cells. Okay? We can have secondary polycythemia. Talk about people living up maybe in the hills when altitude's high, they're gonna have more red blood cells in their blood. Right? It could be a cancer too. Okay? That's secondary polycythemia that is when we live up in the mountains, for example. A cancer of the bone marrow, another cancer, not leukemia, but here it's producing cancerous red blood cells. That's polycythemia vera. Okay, here, what do you think the treatment is? We might have to go to chemotherapy, but initially we'll just keep taking blood out. This one we cannot use to donate healthy red blood cells. When we talk about blood doping, can also have polycythemia, right? Remove, store, their blood body will produce more red blood cells because now red blood cells counts low, oxygen to the kidneys and everything else is low, kidneys release erythropoietin and stimulate that movement also produce additional red blood cells. So polycythemia is excess erythrocyte. Right. Let's talk about leukocytes next. So leukocytes are the least numerous out of the three. Okay? There are 5 different cells. They are divided into 2 groups: granulocytes. These are ones that have got visible granules, and agranulocytes. The granules are not so visible. They do have granules, but not as visible. Hopefully, you'll see a terminology similarity, right? What are the agranulocytes have in common, name-wise? Agranulocytes have the ending sites, right? While the granulocytes
Pills.
Okay, that's the first thing that helps us distinguish between agranulocytes and granulocytes. Okay? So let's talk about these granules for a second. Oh, here, this is just an acronym that helps to distinguish or remind us of the number of the white blood cells. So it's neutrophils that are most numerous, then it's lymphocytes, then it's monocytes, then it's basophils, and then the last one, then it's eosinophils, and then basophils, the last one. Okay, the least numerous. So less than 1%. This is about 1 to 2%. This is about 3 to 5%. This is about 20%. This is about 7%. So let's talk about them five. Oh, in terms of their common function, they have the capability to leave the blood cells, leave the blood vessels. Okay. When we talk about diabetes, okay, during inflammation, especially the neutrophils will squeeze through the membrane to get into the tissue to help with the immune response there. Okay. They can also go by stimuli, so we talk about chemostaxis, that's chemicals, attraction by chemicals. And the last one, they can be phagocytes. What are phagocytes? Phagocytosis? EPOP, right? So especially neutrophils, they phagocytize epithelial cells, eosinophils, they phagocytize parasites, monocytes, they just basically phagocytize anything, all cells, okay, immune cells. So let's talk about the 5 different types, right? Neutrophils, those are the most numerous. So here we want to be able to distinguish them. So a couple of distinguished factors is the nucleus and the granule. Here, nucleus is 3 to 5 lobes. So it looks like, almost like if you look at the old cartoons, if you watch Tom and Jerry, if you've seen them swimming, they have what looks like the flotation device looks like a bilobed structure. Okay, so those are the different lobes. So here, not the best image, but that's 1 lobe, 2, you can kind of see the attachment here, 3 and 4 lobes here. So it's usually 3 to 5 lobe nucleus. The granules here are red and blue. Some of the stains it looks purple because of it. Okay? They do have red-blue granules. I didn't put it here. Maybe you want to include that. I'll talk about it again in lab. Okay, so red-blue granules. They are, in terms of functions, they are bacterial killers. They basically swallow up bacteria. Okay? So be able to distinguish them, be able to find the function of them. I believe next lab, not for Monday students, but for Wednesday students, is we're starting with blood, I believe. So we'll talk about this again soon. Next Monday students will talk about it. So neutrophils will be bacterial killers. Right? If we have pus, right, that yellowy substance, that has a lot of neutrophils trying to fight that infection. Anyway, next one's eosinophils. Eosin is a dye, makes things red. So these are red colored granules. They have a bilobed nucleus. So here are the 2 lobes. Generally, we do not always see the attachment, but they have 2 lobe nucleus with red granules. Okay. In terms of function, they have 2-part function, or 2 different types of functions. One is their main function as an immune cell. They fight parasites, worms, parasitic infections. Okay. They also have antihistamine. What does that do? What might we take it for? Allergic, yeah, it causes inflammation. Histamines cause or promote inflammation, while antihistamine will decrease inflammation. So these one kind of control inflammation, okay? Kind of dampen inflammation, okay? So control inflammation. So that's our eosinophils because they contain antihistamine. Okay? So 2 functions: parasitic infections, they fight parasites, and they contain antihistamine, so they dampen inflammation. You don't want too much. Then basophils, they are bilobe or sometimes considered kidney-shaped. Actually, it's more correct rather than bilobe. They're kidney-shaped and they have blue, very dark blue granules. A lot of times you don't even see the nucleus because the granules are so dark. Here, basically, you will not see the nice kidney-shaped nucleus here. Okay. These are kidney-shaped nucleus, dark blue granules. They contain 2 chemicals. Once you know the chemicals, then you might be able to state the function. It contains heparin and they contain histamine. What does histamine do?
It creates
Mentioned.
inflammation,
Promotes inflammation. Heparin,
prevents blood clotting.
yeah, it decreases clotting, right? So it's anti-clotting. So it will decrease clotting itself. So these are released when we want inflammation. We don't want blood clot. Not in that area. So that can be released as well. Those are the three granulocytes. What are the three granulocytes? Neutrophils, basophils, eosinophils. The two A granulocytes are lymphocytes and monocytes, which will be monocytes, that's the largest of the white blood cells. They're almost double the size of the red blood cell. All the other three that we've so far talked about, it's a little bit larger than the red blood cell. If you notice here, this is one monocyte, but this is a red blood cell. Very large. It's also a kidney-shaped nucleus, not very visible granules. Functional significance, they are macrophages. They swallow up dead cells and other substances. What do lymphocytes do? B cells, T cells. We'll just learn it by here as they have specific immunity. In terms of their shape and size, they are very similar to our red blood cells. They're the smallest of the white blood cells. And what can we note here? This is a lymphocyte. What do we think about the nucleus versus the rest of the cell?
The nucleus is really big.
It's really big, very little cytoplasm. Sometimes you'll see it like a crescent, sometimes you'll see it like a halo, but very little cytoplasm. These are the smallest of the white blood cells. And round nucleus. But now we'll just learn a specific immune response. Okay, what that means is only one cell will only fight a certain species in a subclass of a bacteria, for example. Right? If it comes across any others, even in that same family, it will not be able to identify. And we'll talk a little bit more about that in the immune system. Okay. But I did want to talk about some terminology with white blood cells, and we're going to stop. Leukopenia, same thing with thrombocytopenia. Penia means decreased number. So leukopenia is a decreased white blood cells, leukocytes. Right? What would thrombocytopenia be?
Decrease in platelets.
Decrease in the thrombocytes. Very good. While if we say leukocytosis, different from leukemia, this just means we have good functioning, increased white blood cell count. Okay, so when you and I are fighting an infection, for example, we would hope for leukocytosis. If I have a bacterial infection, you would see which cell numbers go up. Neutrophils numbers. Okay, so that's leukocytosis. While leukemia is a cancer of blood cells, they do not function the way as white blood cells. Okay. We talk about four if you wish, but two subtypes, myeloid versus lymphoid. They can be acute or chronic. Stop there. Here's an image showing the white blood cell formation. Here's hematocytoblast, myeloid versus lymphoid. Lymphoid will give rise to lymphocytes. Myeloid gives rise to the